Hey friends, this is Dr. Cummings from PLNU. We're talking about how to read a journal article, and we've been working our way through this particular paper from Frontiers in Microbiology. Let's remind ourselves of the title here, Characterization of 4 multi-drug resistance plasmids captured from the sediments of an urban coastal wetland. You can really break down all those key terms. They're packed, and just a few words, really, we can learn a ton about what this paper is all about. What I really want to do right now is focus on the introduction. Now, the introduction to a paper is really important, particularly if the idea that's being studied is new to you. If you have been in this field for years now and you've been reading very, very broadly, you may not need to continue to read, at least in any depth, all the introductions to every paper. You may be able to jump straight to the last paragraph in the intro, which tends to describe the purpose of that particular study, and then get right into skimming the methods and right into the results. But especially when you're new to a discipline, reading that introduction can be an absolute lifesaver in 2 ways. One, it sets the stage for the context for this work that's already been done, etc., and helps you to contextualize this research. And 2, it gives lots and lots of citations, other papers you can go find and continue to read so that your own background knowledge and understanding is growing. And that's important, right? As a researcher, as a scientist, that's important when you specialize in something to really study it broadly. So let's take a look at this particular introduction in this paper from Botts et al. 2017. And I want you to keep an eye out as we read this together for some key things happening here, right? Remember I said the introduction is all about setting the context, right? Going from nothing, right? Going from nowhere, broad, to saying, okay, well, here's what we're going to talk about. Let's narrow it down to bacteria, right? We're not talking about frogs or trees, we're talking about bacteria. And then we're talking about antibiotic resistance in bacteria. And then we're talking about solving problems with antibiotic resistance because of plasmids in bacteria. And what we're going to do is we're going to take science and narrow it down by setting the context until eventually we can identify really clearly the need, like where's the edge of our understanding and why do we need to push beyond that edge? And then we can understand the purpose of this particular study. So think about things like context, need, and then an aim or a purpose as we go through this. So the opening paragraph, really important. They start with this very, very broad statement. Bacterial resistance to antibiotics is ancient. Broad, okay? Very, very broad. Bacterial resistance to antibiotics is ancient. It predates their clinical use to prevent and cure bacterial infections. It's been around naturally since even before we started using antibiotics. However, since the beginning of the current antibiotic era less than 100 years ago, human-induced selective pressures against antibiotic-susceptible strains has led to an intractable rise. Here's the problem, right? Led to an intractable rise in the frequency of resistant populations that are on their way up. There's a need to study this, to understand this better. Global public health is now facing an epidemic of bacterial infections with reduced susceptibility to frontline clinical antibiotics like cephalosporins, fluoroquinolones, leading to therapeutic failure, worsening patient outcomes, increased financial burdens, etc. Okay, so we're starting to really see the need here, right? And now the authors nail it and say, look, this kind of work really needs to be done. Viable solutions are urgently needed, I'm here in the last sentence, to protect the long-term efficacy of the antimicrobial agents that we have come to rely upon so heavily. So we started broad and we started really narrowing it down to the need. But the context is still pretty broad, isn't it? It's still, hey, we got a problem with antibiotic resistance. And so they're going to further then take this issue of antibiotic resistance and start narrowing that down. So they start with chromosomal mutations, but chromosomal mutations can only be passed on to the next generation in a vertical manner, which is going to be relatively slow. By contrast, horizontal transfer of resistance genes by plasmids allows the spread to be very rapid in the same generation. Extensive spread of the resistance phenotype. So what exactly is a plasmid? Let's make sure everybody's on the same page with plasmids. And many of them encode what they need to transfer by conjugation. These are self-transmissible plasmids. Another type you should know about are the mobilizable plasmids. But much of the recent rise in antibiotic resistance, it says, is very likely due to the rapid spread of resistance plasmids by horizontal gene transfer. Okay, so we started with resistance is a big problem, and there's a serious need here because people are dying and it's costing us a fortune. But we can't just go there. We've got to narrow this down even more. So we say that, well, chromosomal mutations are an important part of resistance, but really what might be the biggest problem is this horizontal gene transfer with these plasmids. So we're narrowing the field down, narrowing the field down. It talks a little bit more about other ways that resistance, or ways in which resistance is being selected in the environment. Some previous work focused largely in animal production facilities, etc. Human sewage is documented as a vast reservoir. We're really starting to get an image here of how antibiotic resistance can be in nature and potentially come back and impact the human population and human health. So again, get back towards the end of this next paragraph. Each paragraph should really be a thought of its own. So if you read the first sentence and the last sentence, you can get a lot out of it. Release of these genes into the natural environment by way of sewage has been reported by this lab and others. Okay, so these things are getting out from sewage. So, all right, so we got a problem. Nature's a part of the problem. Sewage is a part of the problem. And we got to find a way to narrow it down and really go after it, right? So we're talking more about nature, different resistance genes found in nature. To understand, here we go. Oh, we're starting to get towards the actual purpose of this study and the need for this particular study. To understand the trajectories by which antibiotic resistance genes spread into clinically relevant bacteria, we have focused our attention on the plasmid reservoirs that carry them. And then here we get into the final paragraph, very, very common in an introduction to start with something like the purpose, the aim, the goal, our hypothesis, etc. Sometimes they won't use exactly that language and you're going to have to read between the lines, but that last paragraph almost always gets after the aim of the study. Here these authors made it nice and clear. The aim of this study was to capture and characterize resistance plasmids from an urban wetland receiving mixed runoff that intermittently includes untreated human wastewater. And then frequently they'll also give you a little teaser of what they found. We describe the complete nucleotide sequences of 4 novel plasmids from this environment and the antibiotics to which they confer resistance. So again, just to summarize, the purpose of this introduction is to set the context, right? We're not in the middle of nowhere, we're in a very specific place. And so we're putting a map out, and we're finding where we are on this map, and we're saying, okay, we're going to now push the edges. We're at the edge of what's known and understood in this particular area, and we're going to push the edges, push the frontier, expand our knowledge in this one area, baby steps further, right? I think of Lewis and Clark when they were exploring the Northwest, right? Every step they took was new information on a map. They were exploring the boundaries of what was already known. But in order to do that, you have to start with what's already known, and that's the role of the introduction. So when you read the introduction, keep in mind context, keep in mind a compelling need to study something, and then the actual purpose of this one specific study. You should be able to get all that information directly out of this introduction.